All right. Thank you everyone for joining us today. We are going to get started now. Welcome to the fourth cBioPortal webinar. Today we are going to focus on group comparison. And I'd like to start with a few logistics. First of all, a reminder, this is the fourth in a series of 5 webinars. All the webinars are being recorded and are posted on cBioPortal.org after the webinar is complete. So the first 3 are all available there. Today's will be posted later today if you want to rewatch it or share it with others. And we do have one more next week, which is going to be a great look at the API and R client. So this will be a slight shift from the first 4 webinars, where we've really been focused so far on using the cBioPortal website and how we can use that to analyze data. Next week, we're going to talk a little bit more about other ways to access the data that we host in cBioPortal via our API and the R clients that we provide. So please join us for that. It's going to be another great one. Other logistics, please ask questions while this webinar is ongoing. Please do so using the Q&A feature from Zoom. We have a bunch of people that are going through the questions as I'm speaking to you. Some questions will be answered directly in the Q&A. Others will be saved for the end, and I'll answer them live for all of us. And if appropriate, we'll save some for future webinars as well. If you still have questions after we're done, please contact us. Email us at cbioport@googlegroups.com. This is the best way to get in touch with us and has the benefit of your question and our answer become a resource for the community as a whole in case others have the same question. And as I said, webinar recordings are available on our website. So the first thing I want to do is just acknowledge all of the team members, past and present, that have contributed to cBioPortal, that have made the amazing tool that we get to use today. You can see that we have contributors from many different institutions. This is an international effort. I also want to acknowledge our funding sources, past and present, who have supported us to allow us to do this work. So I want to start with a quick overview, a few slides just to get us started, and then for the most part, we'll be doing a live demo on the site. So cBioPortal is a platform for exploratory, interactive visualization, analysis, and download of large cancer genomics data sets. The software is fully open source, and it's jointly developed by an international team, as you saw on the previous slide. And we support a public website, cBioPortal.org, where we have publicly available data. And there are also private instances installed at academic and commercial institutes around the world. So if you have your own data, perhaps pre-publication, something you're not ready to share publicly yet, I encourage you to reach out to your institute to see if you have access to an instance of cBioPortal where you can use all of our analytical capabilities with your data. Now, we've talked a lot in the past few webinars about the data that we have in cBioPortal, the different types of molecular data, the clinical data, the different data sources, the annotations that we provide to help you interpret the data. And I want to focus on something slightly different today, which is how we can use all of these different data, all of the different kinds of information, and how we can use them to define and compare groups. So we've gotten a lot of questions over the course of the earlier webinars, questions I've also heard in other training sessions, that I've kind of summarized and generalized onto this slide. We got a lot of questions like, how can I compare samples that have mutations in a gene to those that don't have mutations in that gene? Or perhaps, how do I compare samples that have a specific mutation in a gene to other mutations in that gene? How do I compare samples that have mutations in both of my genes of interest to how do I compare samples with mutations in both to samples with mutations in just one or the other? Or how can I look at the expression of a gene and stratify patients based on the expression of that gene and compare the outcomes in those samples? Or how can I take the expression across multiple genes, for example, high in one gene, low in another, find samples that have both of those features and compare them to all other samples? So these are questions we commonly hear, and there's some common themes to what these questions are really asking, which is, how do I define groups? How can I perhaps refine those groups to really specifically target the samples that I'm interested in? And then once I have my groups, what analyses can I run? And so this is what we're going to focus on today. Once you have groups, what are the analyses that we enable you to do? And how can we define our groups to answer all of these questions? So we're going to start today with kind of a general overview of group comparison, and then I'm going to go through examples to answer each of these questions and hopefully give you the tools to answer any of your own questions, the tools to create your groups specific to whatever set of samples you're interested in and ask the questions that you want to ask. So with that, let's get started on the site. So here we are on the cBioPortal homepage. This should hopefully look familiar to you if you've been following along for the past few webinars. And as I said, we're going to start with kind of a general overview of what group comparison can do. But before I do that, I want to point out one additional thing, which is that group comparison is one of the only features on cBioPortal where there's actually slightly different, small differences in what you can do, depending on whether or not you're logged into the site. So most features on cBioPortal are going to be identical whether or not you're logged in. But there are a few differences in what you can do with group comparison. And I will, throughout this webinar, point out to you when I'm doing something that's unique to being logged in and when I'm doing things that you can always do no matter what. So we're going to start not logged in for the moment. And we're going to start with an example in brain tumors. So I'm going to select this TCGA publication from 2016. This is a data set of low and high grade gliomas of brain tumors, and we're going to start by exploring this study. So this is often how we're going to start with group comparison, is by going to study view. And what we see when we're on study view, we've seen this before, are a series of charts representing various clinical data elements that are available for this study, as well as summaries of some of the genomic data. So most frequently mutated genes, most frequently copy number altered genes. When we're not logged in, so the main difference between when we are or not logged in is in how we can define our groups. So when we're not logged in, we can use any of these existing clinical charts to define our groups and to enter group comparison. So I'm going to show a first example. I'm going to actually start by adding a chart, which is not visible by default, which is the grade of the tumor. And we see it adds here down at the bottom. So if we look at this chart, we can see that the samples, each sample has a grade. About half of them are grade 4. The rest are divided between grades 2 and 3. And so what we can do is we can use these grades as our group definitions. So I can come here to this dropdown, and I have an option to compare groups. And what this is going to do, I'm going to click it, it's going to bring us to group comparison with groups defined based on this clinical element. So we have 3 groups, grade 2, grade 3, and grade 4 samples. Now that we're in group comparison, everything that we're going to see, this is all now going to be identical whether or not we're logged in. What we're going to see is an overlap tab. This is where we're always going to start. And it's going to show us if there's any overlap among the samples or the patients in our groups. So here you'll notice those 2 charts, the 2 overlaps look identical. If I jump back to study view for a second, we can see that in this study, there are the same number of samples in patients. So every patient has exactly 1 sample. There are some studies, and we'll see some later today, where some patients have multiple samples. And when we're in that scenario, you might see a difference between your sample overlap and your patient overlap. But so we'll always start here where we can see if there's any overlap among our groups. In this case, there is not. And then we have these additional tabs we can work our way through. So when there is outcome data available, and not all studies have outcome data available, so you won't always see this, but when we have the data, you'll see a survival tab where you can compare the outcomes among your various groups. So we can see here, how do patients with grade 2, grade 3 versus grade 4 tumors do? We can see quite clearly that patients with lower grade tumors do have better outcomes. We can continue along and look at the clinical tab. So this tab will take all the same clinical attributes that we saw in study view. All of those show up here with a statistical comparison to see if there's a difference in that attribute among all the groups. So we can click on, for example, this methylation clustering. We can see, once I click on it, we see the chart on the right shows us that information. So we can see that there is pretty clear difference in which methylation clusters are present depending on the grade of the sample. You can customize what this chart looks like. We give you a few different options for the plot type, for kind of swapping your X and Y axis. And like everything else here in cBioPortal, the plot is downloadable, the table is downloadable, so you can always take this data for later. I did mention this is a statistical analysis. If you're ever confused about what statistical analysis has been done anywhere where we show a p-value or a q-value, you can always hover over the column name. It'll tell you exactly what test we've done. In this case, the test is actually a column all its own, but you can always confirm that via the tooltips that will appear in the column headers. So this is what the clinical tab is going to look like. We then have a series of tabs for each available molecular profile. So this study only has mutations and copy numbers, so that's what we see. If we had RNA expression, if we had protein expression, those would appear as additional tabs, and we will see that in some later examples. But all of these molecular profile tabs do a similar thing, which is to say, are alterations in each gene enriched in one of our groups? So on the mutations tab, we're asking, are mutations in ATRX enriched in one of our 3 groups? Are mutations in IDH1 enriched in one of our 3 groups? And we can see, again, we have statistical calculations to support that here on the right of the table. We can see what test is being applied. And we can see that there are, in fact, significant enrichments. These first 2, ATRX and IDH1, are significantly enriched in the grade 2 samples. We can see that same information represented in the table, where for each gene we're plotting, or in this graph, where each gene is plotted along with the frequency of mutations in each of our groups. You can actually modify this graph to show any genes of interest. Now, right now, this is what we see when we have 3 or more groups selected. We can always change which groups we're looking at. So I can always choose to deselect a group. I'm going to deselect grade 3 for the moment. And now this analysis is rerunning, recalculating, so that we just compare the grade 2 and the grade 4 samples. And when I do that, when I have only 2 groups selected, we'll see these 2 additional graphs on the top. This one is showing us the frequency of alterations in one group versus the other. We can see here, so we can see in the table below, there's been a change. IDH1 is now the most enriched. We can see that here as kind of the most significant outlier on this graph. This is a volcano plot, so we're plotting the significance against the enrichment. We can find, again, IDH1 represented as one of the most significant genes. So this is the kind of thing you can do once you get to group analysis. You can look at whether or not there's overlap among your groups. You can compare outcomes, you can compare clinical attributes, and you can compare all the different molecular profiles that are available. And you can, at any point, choose to include or exclude a particular group from your analysis simply by clicking on it. If you don't have at least 2 groups, you can't do a comparison. So this is what we can do in group comparison. What I'd like to do now is go back to study view and show you one other way we can define groups when we're not logged in. And then I'm going to log in and show you what we can do in that scenario. So what we did right now was we took grade. And when we look at the grade pie chart, we can see every sample falls into one of 3 groups. It's pretty easy to think about how you can define groups based on a categorical variable like this. But what about something continuous? What if we take something like diagnosis age, where every sample has a different value, and we want to compare groups based on that? So we can do that, and we actually have a few choices about how we do that. So once again, I'm hovering over this little menu icon, and I still have a compare groups option, but here, this is actually a set of choices. So I can define groups in multiple different ways for a continuous variable like diagnosis age. I can define groups as quartiles, so this would be 4 equally sized groups. We can do based on the median, so 2 groups above or below the median age. Or we can define groups based on the current bins in this histogram. You actually have the option to define your own bins in any histogram. And if you do that, you can then go to group comparison based on your defined bins. So as an example right now, let's go through to group comparison based on quartiles of diagnosis age. So when I click here, it brings us back to group comparison. We're going to see all the exact same tabs, all the same analytical options. This time we have our 4 groups. We can see 4 groups of equal size. And we can ask all the same questions. So are there differences in outcome based on the age at which individuals are diagnosed? And there certainly do seem to be. Younger patients seem to do better than patients diagnosed at an older age. We can look at clinical features and say, are there differences in various clinical features among these groups? If we click on age, we can see quite clearly that patients diagnosed at an older age tend to have higher grade tumors, perhaps related to the worse outcomes that we saw on the survival tab, etc. So you can continue through and ask, are there genes that are mutated more frequently in one group or another? Are there genes that are amplified or deleted more frequently in one group or another? So this is what we can do when we're not logged in. So let's come back to study view now, and let's talk about other ways we can define groups. So there is this groups dropdown that you might have noticed here on the page. And while I'm not logged in, this is actually disabled. So this is the major difference between being logged in and not being logged in. Let's log in and let's see what we can do with this groups dropdown. So logging in, Very simple process. Anyone can register. Just takes a Google account or a Microsoft account. Very straightforward process. And now I'm logged in with my account. And we can see everything looks the same. But when I click on this groups dropdown, it's now populated. I can use this. And what this lets me do is it lets me define my own groups. So I can define groups by filtering study view any which way I want, by any single chart, combination of charts. I'll show you how you can actually paste in your own list of samples, any way you want. And at any point, as you filter study view to a group of samples of interest, you can create a new group from those selected samples. And these groups that you create are saved to your profile. So the next time you log in, those same groups are going to be present. You can use them over and over again. And you can define many groups, and you can choose which among them you want to compare at any point. So this is a really powerful way to really define groups of interest to you. So I'd like to just show you very quickly what we can do, kind of building off the same example, which is to say, let's look at diagnosis age again. But instead of comparing everyone, let's just take the oldest and youngest patients. So we can do that by using this graph to filter down. So I'm just selecting the kind of bins that I want to include. So I've selected everyone diagnosed at age 60 or above. We can see all charts on the table on the page are updating because we're actually filtering to just those samples. And now I can come to my groups dropdown and I can create a new group. We'll call it age greater than 60. And now this is saved, and every time I come back, this is going to be here. So that's our older patients. Let's create a group for individuals diagnosed at a young age. So I'm going to select everyone under 35. And I'm going to create another group for age less than 35. And now that I have two groups, again, I could make more. We're just going to start with two right now. I can compare them. And so when I click compare, I'm coming back to the exact same group comparison we've been in before. Same tabs, same analytical features, but with groups defined my own way. So we can look again at outcomes. We can see, as we saw before, younger patients diagnosed at a younger age do better than those at an older age. And we can do, again, all of these same things that we've done already. So with that kind of general introduction to what group comparison can do and kind of some basic ways we can define groups, I'd like to continue along and do some examples that address those questions that I laid out in our slides. And in doing so, show you additional ways that we can use group comparison, additional ways we can define our groups. So I'm going to come back to the homepage and I'm going to run a new query or do a new example. And in this example, I want to take up the question of how can we compare patients or samples that have mutations in a gene to those that don't have mutations in a gene. So we're going to do this example in pancreatic cancer. We're going to take this legacy TCGA study, and we're going to come on back to group comparison, to study view. And what I'd like to do is ask the question, what are the differences that we can see between samples with or without mutations in KRAS? So we can see in this mutated genes table, KRAS mutations are very common. 90% of these pancreatic adenocarcinoma samples have a KRAS mutation. What's going on in the samples that don't have one, and how does that compare to the samples that do have a mutation in KRAS? So in order to answer that question, I need to create two groups. I need to create my group of KRAS mutant samples, and I need to create a group of non-mutant samples. So the KRAS mutant group is pretty straightforward. I can filter study view to samples with KRAS mutations. Again, we see all the charts are updating because we're applying a filter. And now I can create a group. So using my groups dropdown, I can create a new group, which is KRAS mutant, and save that group. So there's my KRAS mutant group. What about creating a group of samples without KRAS mutations? So there's actually a couple ways we can do this, and I'm going to show you two of them. One way that we can do here on study view, and one approach that we can take using group comparison to define our groups. So to start with the approach here on study view, to do this, we're going to take advantage of this custom selection dropdown. Now this dropdown does three things. First thing it does is, you can see we have a nice box here. In this box, we can enter a list of sample or patient IDs. And this list of sample or patient IDs can come from anywhere. It can come from an analysis in cBioPortal. It can come from your own offline analysis. As long as you enter the kind of the same IDs that we're using, you can enter a list here and you can filter down to those samples. And from there, you can create a group if you want. You can just explore the samples in study view, whatever you'd like. So that's one thing we can do here. Another thing we can do is use this to actually get the list of samples based on our current filter. So there's this currently unselected option here. What that does is it populates the text box with all the samples that I've filtered to. So right now, this is a list of samples with a KRAS mutation. But that's not what I want. I already have that list. What I want is the currently unselected samples. So these are the samples that don't have a KRAS mutation. The samples that I've filtered away, so to speak. This is the set of samples that I'm looking for. So I can use the currently unselected to get this list of samples, and then I can apply a filter to just those samples. And I can prove this to you. If we look at the mutated genes table, I can search for KRAS. Nothing is found. There are no samples with KRAS mutations left. So great, we've got a set of 50 samples that don't have KRAS mutations. But there's one other thing we have to think about first. Anytime you're doing an analysis that says, give me samples without something, there's something you need to think about, which is, did we look for that in the first place? So given the 50 samples that we currently have, did we actually try to call mutations in all 50 of them? Because if we didn't try to call mutations, we actually don't know if they're truly not KRAS mutant or if they are KRAS mutant and we just didn't see it. So the way that you're going to want to do this is to use this genomic profile sample counts table. So this shows us all the genomic profiles that are available for this study and the samples that have each profile. So we can see that of the 50 samples that we're looking at right now, only 14 of them had mutation profiling done. So it's really these 14 samples that we want to use as our KRAS not mutant group. So what I can do is I can apply another filter, get to just samples that had mutation profiling done. And now I can create my group, which is my KRAS no mutations group. So I'm actually going to change the name of that. So you can see once you create a group, you can edit it, you can delete it, you can actually add samples to it as well. I am just going to edit the name real quick and say this is the group we made on study view. Oh, that was not what I meant to click. I meant to click save. So I told you there's two different ways we can create this group. This is the group we made on study view. Let's do what we can. I'd also like to show you how we can create a group using group comparison. So in order to do that, we do need to define one additional group here. And that again is our background set of samples. It's our samples that have mutations profiled. So I'm going to create a group. I'm going to filter down to samples with mutations, and I'm going to create one last group, which is samples that have mutations. And now with these three groups, let's continue on to group comparison. So again, remember that one of the nice things about group comparison is we can kind of select and deselect groups at will. So I'm going to just start by deselecting everything. And I'm going to show you how we can create this group again, but create it from group comparison. And so to do that, we're going to take our has mutations group and we're going to take our KRAS mutant group. And we see here now on the overlap tab that these are overlapping groups. They're in fact, one group is a subset of the other. And you might notice that this KRAS mutant group is kind of grayed out on the top. It's because that group is a subset of the has mutations group. So once we get to a scenario where we have overlapping samples or patients, we need to decide whether to include or exclude those overlapping samples and patients in our analysis. So you actually have a choice. You can control this. By default, we're going to exclude them. We can choose to include them, in which case you see this group kind of comes back to life. But when we exclude them, any group that is a subset of another group is going to be kind of grayed out, because what that means is that it's really not available for doing any comparisons. You can see all these tabs are grayed out because we kind of only have one group to work with. Even when they're grayed out here, you'll still see them in the overlap tab. All selected groups will be shown here, but other tabs might be disabled or not taking that into account. So we set out to create a group which is samples that have mutation data but not KRAS mutations. And if we look at this overlap, that's essentially these 14 samples, right? It's the samples that are in the has mutations group and not in the KRAS mutant group. So what I haven't shown you before is that when you're on the overlap tab, you can actually use this tab to create new groups. So I clicked on this part of the Venn diagram, the part that I'm interested in, and now I have this button down below to create a group from the selected areas. When I click this, I'm given an option. I can enter a group name. So this is KRAS no mutations, but now from the overlap tab. And this you can do whether or not you're logged in. Because I'm logged in, I also have this choice to save the group to my user account. So again, creating the group I could do when I'm not logged in, but saving it to my account, I only can do that when I am logged in. And what this means is that when I go back to Study View, this group is going to be saved to my profile, just like the other groups that I made on Study View. So this group becomes something I can come back to later. I can use it again in the future without having to recreate it each time. So let's create this group. And then just to kind of prove to you that we've done the same thing in two different ways, I'm going to select both of my no mutations groups, and we can see that they're perfectly overlapping. We found the exact same 14 samples in two different ways. Great. What we really want to do though is do the comparison, right? We want to take our KRAS mutant group, we want to take our no mutation group now that we have it, and we want to compare them. So we want to ask, are there differences in outcome? Not significantly. We can see here, this study has both overall survival currently shown. We can also look at disease-free survival, also in this case, not significant, and we can continue along. So this study has RNA data. It has RNA expression and protein expression. So we can look at this and we can ask, are there genes that have higher expression in one of my two groups? That's what we're going to do on the RNA tab. Similarly, the protein tab will ask, are there genes that are have higher protein expression in one group or the other? We can see in this case there's nothing significant, but we can always continue to explore the data some more. So this is an example of how we can use both study view or group comparison to define groups of samples with or without a particular event, and how we can kind of use group comparison to strategically combine groups to create the groups that we really want for our analysis. I'd like to continue along with another example, kind of building on this one. Here we asked about the presence or absence of a mutation in general. What if you want to talk about a specific mutation? So we're going to do an example in colorectal cancer. And what I'd like to ask is, what are the differences between BRAF V600E mutant samples and other BRAF mutations? Are there differences? What can we learn from that? So to do this, I'm actually going to run a query. And I'm going to take advantage of OncoQuery language, or OQL. And you were introduced to this briefly in the last webinar. I'm going to kind of cover it again today. So I'm going to start just by querying BRAF on its own. And what we see when we query a gene, what we see when we're going to query BRAF right now, are a variety of different types of alterations. We see fusions, amplifications, and mutations. To take a step back, when I tell cBioPortal I want to query this gene, what types of alterations am I going to see? Am I going to see everything? So we make an assumption when you query a gene. We kind of decide by default to show you amplifications, deep deletions, mutations, and fusions. And on the topic of fusions, I do want to point out, not all studies have fusions, many don't, in fact, so you often don't see them simply because the data is not there. But when the data is available, you will see them. These are the 4 data types you're going to see. This is what happens by default. The power of OQL is that it lets you define exactly the types of alterations that you want to see. So I can come back and modify my query, and I can use OQL now to say I only want to see mutations. So anytime you run a query, you're going to see this link here. This is a link to a page, full documentation on how to use OQL. We also have a tutorial on the topic, so you can refer to those at any point for kind of reminders and the specifics of how to do it. But the basic way of using OQL is pretty straightforward. You take your gene, you follow it with a colon, and then you enter the keywords for the types of alterations that you want to query. So in this case, I only want to query for mutations. So I say BRAF : mut, and I can submit that query. And now back on OncoPrint, we see only mutations, no more amplifications, no more fusions. We're only looking at mutations. So that's great, but that's actually still not what I want. The question I wanted to answer was, how do samples with BRAF V600E mutations compare to samples with other BRAF mutations? So we can do that with OQL as well. So I'm going to come back and edit my query again. And this time, I'm going to say, give me not just any mutation, but give me specifically V600E mutations. And when I submit that query, we're now going to see two tracks in our OncoPrint, one of all mutations, and a second, which is a subset of the first, just the V600E mutations. So we can use OQL to identify subsets of alterations within a gene, specific mutations, specific types of mutations. There's a wide range of things you can do with OQL. I encourage you to look at the documentation and to review the tutorial on the topic for a full understanding of all the things you can do with it. For right now, though, I'd like to focus on this question of V600E versus other mutations. How are we going to go from running this query to actually defining those groups and comparing them? So what we have when we run a query is we have a comparison tab. And this comparison tab is actually identical to the group comparison that we've been using when we get to it from StudyView. All the same tabs, all the same functions. The only difference is in how your groups are defined. So when you use comparison from a query, you're going to start by default with two groups selected, altered and unaltered samples. So your altered group is any sample that has one or more alterations in one or more query genes. So any alteration at all, it'll be in the altered group. The unaltered group is the group of samples with no alterations in all of your query genes. So fully no mutations, no events. If we look on OncoPrint, it's essentially everything to the left of my mouse is altered, everything to the right is unaltered. Nope, comparison. So that's great, but that's actually not the question we want to ask. What we see is that in addition to the altered and unaltered, we have groups, currently unselected, but groups that reflect each of the tracks in OncoPrint. I'm going to select those, but before I do just want to point out one other thing. We're looking at a study here that has more samples than patients. So some of the patients in this study have multiple samples that have been profiled. And we can see that in these two overlaps where we see there's actually a difference now between the samples and the patients. It appears that there's one patient that has at least two samples, where one sample is altered, one sample is unaltered, and so we have this kind of overlap here that we don't see at the sample level. So this is kind of what you'll see when you have more samples than patients. But to get back to my original question, what I really want is to compare V600E to all other mutations. So let's look, look at the Venn diagram, looking at the V600E and the other. And as we saw before, we can use the overlap tab to define a new group. So here's my V600E, I already have that group defined. This 48 samples that are in the BRAF mutant group, but not the V600E group, that's the group I want to define. So I can select it, I can create a group, we'll call this BRAF V, not V600E. And again, note that I can save this back to my account. So again, if I were not logged in, I could still create the group. Because I'm logged in, I have the additional option to save this to my user account, which means that the next time I come back, this group will be saved in StudyView, and I can use it again and again for future analyses. So let's create this group and save it. So now we have the group we wanted, and we have our V600E group. Deselected the wrong thing. There we go. So we have our V600E group, we have our not V600E group. There's one other thing I'd like to do before I continue to actually doing this analysis, which is actually to resave this V600E group. So when we save a group, when we kind of create a group, we can save it back to our profile so that we have it for the future. Right now, this group only exists in the query. I can still select it even though I'm essentially creating a duplicate group, and I can create a new group. And we're going to call this V600E, and we're going to save it back to our user account. So here in the context of this query, it's essentially a duplicate group, but by kind of recreating it and saving it, I've saved it for myself for the future, so I don't need to run this query again the next time I want to ask the same question. So having done that, I now have my groups, I can do the comparison. So we can ask, are there differences in outcome in colorectal cancer and metastatic colorectal cancer, just based on what kind of BRAF mutation you have? And indeed, there are significant differences. We can continue along. We can ask if there are clinical features that are different. We can ask if there are genes that are mutated more commonly in the presence of a BRAF V600E mutation or in the presence of a BRAF mutation that is not V600E. And so we can see that indeed there are KRAS mutations. Interesting, because KRAS is often considered mutually exclusive with BRAF. KRAS mutations are preferentially found when there's a BRAF mutation that's not V600E compared to when there's a BRAF V600E mutation. So you can continue through, as you can see, and ask all the same kinds of questions we asked in group comparison from StudyView. We can do it here from running a query, using the query to define our groups of interest, and again, using the overlap tab to refine how we, to make new groups to ask the questions we want to ask. So this is one way we can use a query to define a group, is using the overlap tab to create groups. I want to show you there's actually another way we can use a query to define a group. And to show you that, I'm going to actually modify the query, delete the BRAF mutant track, and just query for V600E mutations. So you might notice this is kind of becoming a common theme. There's often multiple ways to do the same thing. And I'd like to try to show you some of the different approaches so that you can use them as kind of fit your questions best. So just to come back to OncoPrint real quick, we can see we're only looking at BRAF V600E mutant samples right now. So what is this other way that we can create a group? Instead of using the comparison tab, let's come over to the download tab. And here you have options to download data associated with your query in a variety of different ways. The one I want to focus on is this list of altered samples. This is all the sample IDs of any sample that has an alteration in any query gene. I can copy this list, and now I can come over to StudyView, and I can use that list to filter StudyView and to create a group. So we can come back to this custom selection dropdown, and we can say, I can paste in my list, I can filter to those samples, and I can create a new group. So this is another way, and again, you can paste in a list of samples from anywhere, but I wanted to show how we can use a query to get a list of samples that we can paste in here and use it to create a new group. Note, we'll call this again, that the other two groups that I created back when we were in the query on the comparison tab, those groups appear here because I saved them back to my profile. So I have them available for future analysis as well. So far, I've shown you a couple different ways we can define groups, how we can use the overlap tab to define groups, but I've so far been very focused on mutations. I'd like to focus a little bit now on RNA expression and other types of expression and how we can use that to define groups. So a really common question is, can I take expression of a gene, stratify patients into high and low expression, and compare those? So you actually saw an example of this in the last webinar, in the expression-focused one. And again, there's two ways we can do this. You saw an example of one of them last week. I'm going to refresh your memory of that real quick, and then I'm going to show you another way that we can do it. So for this example, we are going to start by querying this legacy TCGA study. So this was the way you saw it done last week, which is you can use OQL to say, give me samples that have high expression in my gene and give me samples that have low expression. Remember, if you're going to use expression in your query, you're going to want to select RNA as a genomic profile. And an important thing to point out here is when you do it using OQL, using a query, you're setting cutoffs based on a z-score. So this is give me samples with a z-score greater than 1 and a z-score less than -1. So I can submit this query, and as we've done before, I can come straight to the group comparison tab. My altered group, of course, is samples with high or low expression. My unaltered group is everything in between. That's not the samples, that's not the groups I want to compare. So I can deselect those, select my individual tracks, and now I can compare and I can say, are there differences in outcome between samples with, or patients with high or low expression of EGFR in this example, etc. You can continue through all the tabs. So this is one way to do it using OQL. What I would like to show you is another way that you can do it from StudyView. So we're going to go to StudyView for the same study. And what we've seen on StudyView already is we have charts of clinical data, we have charts that summarize mutations and copy number, but there's no real summary of expression data. What we let you do instead is we let you add expression of genes on an individual basis. So here in this charts dropdown, where we can add clinical charts, there's a gene-specific tab. And here, you can enter any gene symbol. So we're going to start with CIC, and you can select any of the available molecular profiles. So this study has protein expression, RNA expression from microarray, RNA expression from sequencing, linear copy number, DNA methylation, all of these different continuous variables, all of these are available to add. So I explicitly want to point out that you have the option here to look at z-scores or to look at the non-z-score transformed data. So again, when you run a query, when you use OQL, you can only use z-scores. If you want to do it based on the underlying data, you can do that here. I'm going to add z-scores though. So I'm going to add RNA-seq z-score. So I'm going to add a graph of CIC z-scores. We can see it's added. It adds down at the bottom of study view. And here we have now a histogram of CIC z-scores across all samples. And now, just like any other histogram, I can use this to define groups. So I can use the dropdown to compare groups in these various ways, or I can manually select. I can say, give me samples with the highest expression of CIC, apply that filter. Oh, I included NA, so let's reset. Didn't mean to include NA in that. So I will select just the high expressing, not the NA, and I will save that as a group. So we'll save this as a group called CIC Z greater than 1. And now I can create kind of the complementary group. I can create the group of samples with low CIC expression. So let's take everything under -1. And I can create another group from there. And we can say CIC z-score less than -1, create that. And now we can take these two groups defined here on study view and go compare them in group comparison. So we can see, again, all the same functionality. Are there differences in outcome between sample patients with high or low expressing CIC? Not in overall survival, but indeed there are in terms of disease-free survival. Are there differences in what genes are mutated? Are there differences in the expression of other genes in this group that's actually defined by expression of one particular gene? And what we're going to see when this loads, kind of a nice sanity check, is that CIC is actually the most significantly differentially expressed gene, which is really what we would hope to see, given that that's how we've defined our groups. But there's lots of other genes that would certainly be interesting to explore. So here I've shown you how you can create groups based on one gene. Let's come back to study view. And all of the same principles apply if I want to essentially combine expression in multiple genes to define my group. So let's add charts for EGFR and PTEN expression. And again, these are going to add down at the bottom. So let's clear my current filter on CIC and let's create groups. Let's compare samples that have high EGFR expression and low PTEN expression. Let's compare those samples to everyone else. So we can select our high EGFR samples and create a group from those. So EGFR high. And then we can clear that filter. We can come on down and we can select our PTEN low sample, so PTEN less than, z-score less than -1, let's say, and come on back up and we can save this as a group. And so you can probably already envision how we can use group comparison to combine these two groups to create that set of samples that are EGFR high and PTEN low. But what I want to do is compare them to everyone else. So we need to define that everyone else group. And again, what we want to do when we define the everyone else group is restrict it to samples where we've looked for this particular data type. So I want to only take samples that have expression z-scores available. It turns out that's actually every sample in this study, but it's an important step to take to make sure you're using the correct background set of samples to compare against. So I'm going to create one last group, which is essentially samples that have expression z-scores. And now I'm going to deselect CIC. I don't want that in this comparison. I'm going to compare my EGFR high, my PTEN low, and my everyone with expression. And I can now create my group of interest, which is samples that are EGFR high and PTEN low. I can name this group and save it. Again, saved back to my user account, so it'll be there for me later. And then I can create my other group. So I can select my everyone that has data group, my group of interest, and I can select the non-overlapping section and call this not high EGFR PTEN low. Save that group, and now I can do my comparison. So I can compare EGFR high and PTEN low to everyone else, and I can ask, are there differences in outcome? Indeed there are. Are there differences in clinical features, mutations, etc.? I am very much running out of time, so I'm going to stop there and take questions. Hopefully this has given you a good understanding, a good feel for how you can define your groups in many different ways, using queries, using study view, and how you can use the overlap tab to really strategically combine groups to get at the sets of samples that you're interested in. So yeah, with that, I will pause and we can answer some of the questions that have been coming in.
Thank you so much, Tali. It's obviously a lot you've covered here. I think there's probably no harm in showing some examples, again, using maybe slightly different examples. As everything, we've experienced it a lot. The more you see cBioPortal and use, the clearer things become. One question that a lot of people seem to have, which might be something we need to fix a little bit. Actually, you're looking at an example right now. What does it mean when the log rank test p-value is 0.0? Is that an error, or is that actually a meaningful result?
Thank you for that question. I actually meant to comment on that. That's a great question. So what this actually means is the p-value is super small. So it's essentially so small that it's gotten rounded down to 0. You will see this sometimes also in, I don't think this example may not have it, but sometimes you'll see it also in these charts, and you'll always see it's kind of ranked at the top of the list. It is essentially the most significant, where it's so small that it's been rounded down to 0. So it's very significant is what that means.
Yeah, and these comments are very helpful for us to help us improve the portal, so we will probably make that clearer in the future. Yeah. A nice feature request that came in, people asked, hey, can I change the color of the Kaplan-Meier plots if you don't? And that's a feature that we're considering and we'll hopefully add soon. That would be nice to have. People asked about combining groups. Let's say you have a study where you have grade 1, 2, 3, and 4 tumors, and you can separate them nicely into 4 groups. But what if you wanted to compare grade 1 and 2 together against grade 3 and 4 together?
Absolutely. So let's come back to this same study, and let's see if this is a study that has grade as an option, and we can go ahead and do that. So this is definitely something you can do. So the same way that I've been using the overlap tab on group comparison to essentially pull out subsets of groups, we can combine groups in the exact same way. So, well, this is going to be a little silly since we only have 2 groups, but it will serve the purpose. So we can combine. So I can select multiple regions here on the Venn diagram and create a new group, which is the combination of all of them. So I could create a group here, which is grade 2 and 3. Obviously, this is more useful in an example that had more grades, but you can always combine pieces as well. So I can always select this and this and this if I want, or this and just this. Any combination of sections you can select and create a new group from those. And actually, just to be clear, you might notice we sometimes switch on the overlap tab between showing you a Venn diagram and showing you what's called an upset diagram. So you're going to see the upset diagram generally when 4 or more groups are selected. And you can select subsets on here just the same as you can on a Venn diagram. So in any kind of visual orientation of the overlap tab, you can select any number of subsets of or complete groups and use them to define new groups.
So you're showing some graphs here. Can users use these graphs in publications?
Absolutely. Please do use these graphs in publication. Every graph that we have on the site is available for download. and it's available for download as SVG or PDF so that it's really easy for you to, adjust the text, change the font, whatever you need to do to make the graph kind of fit into a presentation, fit into a publication. The only thing we ask, if you are going to use data or analysis that came out of cBioPortal, is to please cite us. So here on the homepage, we list our citations. And please cite the study that generated the data if it's not your own data. So every study that we list, we have a link to the PubMed citation of where that data came from. So please do cite that as well as us when you use data analysis graphs, anything that comes out of cBioPortal.
Great. A lot of users seem to be interested in, again, separating samples based on expression levels, often of expression of one specific gene. So TCGA breast, for example, we can maybe pull up the Cell manuscript and show users how to stratify by ER high versus low patients based on the mRNA levels of ESR1, or maybe even the protein levels of, if those are available in that particular study of
Absolutely.
ER.
I think if we do the legacy, we should have protein data. So we can do this in two different ways. We can do it by running a query and using OQL, or we can do it in StudyView by adding charts that show the expression of individual genes and defining groups based on that. So here we are in StudyView, and we can come to the charts dropdown. We can add a gene-specific chart. So we can add it for the estrogen receptor, ESR1, and we can select which type of data we want to see. So we can do this based on protein expression, the actual expression values, or the Z-score values. We can do it, this is based on RPPA. There's also expression data from mass spec, from CPTAC. We can add that data in kind of its raw form or its Z-score form. We can add it as expression data from microarray or the Z-scores, as well as from expression from RNA sequencing and the associated Z-scores. Let's actually do this based on the protein expression Z-score. So I can add this chart. It's going to add as a histogram. It's going to show up at the bottom of the page. And we see here what the expression distribution is. The Z-score distribution is a histogram of the Z-score in every sample in this study. And from here, I can select, for example, my lower expressing samples, selected just by highlighting it on the graph, and then we can come up to the groups drop down. We can create a new group, which is ESR1 protein low. And we can clear that filter. We can define another group, which is the high. So let's take everything up to, but not including the NA group. And we can define another group, which is our ESR1 protein high. And we can now compare these two groups. So we can click through to group comparison, and we can see, are there differences in outcomes? Are there differences in clinical features? Are there differences in mutation, copy number, etc., among these different groups or between these different groups? So we can see overall survival. We can see disease-free survival, not significant in this case, clinical features, etc. We can see, actually, conveniently, ER status by IHC is significantly different between our groups and generally seems to correlate with our definitions based on protein expression. So that's actually really nice to see that this is kind of concordant with other methods of measuring the same thing.
Yeah, and of course you can use a first selection, basically. For example, once you've identified your ER positive samples, when you go back to the study, you could then, of course, layer on top of that a gene expression comparison for another gene.
Absolutely, and we could also look at that ER. I forget now what it was called. ER positivity by IHC, you can look at that here as well. You could also use that to compare your groups if that were of interest to you.
Maybe you can show how to do that, maybe ERBB2 in ER negative or something. I think that question showed up a little bit.
Sorry, to look at ERBB2 expression in ER low samples?
For example, yeah.
Sure. So one of the fun things about defining groups is you can not only use them to go through to group comparison, you can actually use them to filter study view. So once you've kind of created the group, I can reapply that estrogen receptor filter. So I can look at just ESR low samples based on this protein cutoff. And I can add a chart for ERBB2 expression. I'll stick with protein for the moment. At protein z-scores to be consistent, we can add this chart, and we can now look at what is the distribution of ERBB2 expression, protein z-scores, in samples that are estrogen receptor low, because recall we've already applied this filter. For the low samples, we can see what this looks like. And we could further define groups if we wanted to based on ERBB2 expression and compare those if we want to in group comparison as well. So that, I think, does that answer the question?
I think that just answers the question and shows how you can layer selections onto each other. So you've now created a lot of groups in a lot of different studies. Are they being saved? Can you get back to them?
Absolutely. So they are saved. They are here. So what we can do is I'm actually going to log out real quick, and we're going to log back in, and you will see that all the groups that I made, they're all still there. So if I now re-log in, sign in with Google, we can go back to one of those studies. So let's see, we did this colorectal cancer study. Let's go back to study view in this study. This is where we looked at BRAF V600E versus not V600E. If I come back to my groups dropdown, just taking a moment to think. There we go. We see all my groups are right there waiting for me. I could immediately jump back into that same comparison that I was doing before.
There's some information available also on how these were generated, correct?
I think it's just the names of them.
Okay,
No.
so sorry, I'm mixing it up with the virtual studies. Virtual cohorts have that information. And then, so it's up to you how to name those groups. And you can delete them if it gets too unwieldy, you can remove them and then they'll be gone.
You can delete them. You can also change their names if you decide you need to make them a little more clear or something. And you can add samples to them as well. If you kind of want to build a group in pieces, you can use the little plus sign to keep adding samples to a group.
And then maybe one last question that I can just answer quickly was about p-values. Sometimes survival charts don't have p-values, and the simple reason is that we currently only support p-values for comparisons of 2 groups, but not 3 groups or more.
Yeah.
We will consider adding p-values to that, but we would have to add one combined p-value and then probably all p-values for each pair. Something we could do.
Yeah, so just as an example, here's a very randomly chosen feature that has 3 different groups, no p-value. As soon as I deselect any one of those groups, we'll get a p-value back. So you'll see it with 2, but not with 3 or more.
Great, thank you, Tali.
Okay, I guess we are out of time. So thank you everyone for joining us. Again, this will be posted online later today as a reference if you want to come back and watch it, along with all of our previous webinars. And we have one more next week, and we hope to see you back then. So thank you all for attending.